Oh, no.
So, who's going to be the first person to break the streak? What streak? Got a week of classes now. No one's missed yet. For your surgery, maybe? We don't count that. Okay, anybody have any questions? Okay, we're done with our introduction and our kind of little history lesson. And so we're now ready to start diving into some of the content of microbiology. So we've got 3 domains according to the newly accepted classification system. How do 2 of those 3 domains differ from the other? They consist of cells that are prokaryotic in nature, okay? In the old 5 kingdom system of classification, all of those prokaryotes were lumped into the kingdom Monera. But now, gene analysis is indicating that there are fundamental differences between prokaryotic cells that we now call archaebacteria and the regular bacteria, if you like. But they're all prokaryotic in nature. So let's do a little review. What are some of the fundamental differences between prokaryotic cells and eukaryotic cells? Parker?
Membrane-bound organelles.
Okay. Eukaryotic cells have the membrane-bound organelles. Prokaryotic cells don't. Are ribosomes considered to be membrane-bound organelles? No, they're both in prokaryotic.
Different ribosomes.
But they have different ribosomes. Okay, good. We have different S values of those ribosomes. We'll talk about that a little bit more later on. So, membrane-bound versus non-membrane-bound. What are some other differences prokaryotic and eukaryotic?
The nuclear envelope.
Okay, yeah, one of those membrane-bound organelles that typically defines a eukaryotic cell is the membrane-bound nucleus. That's what the word eukaryotic really means. Eu refers to true, and karyon refers to nucleus. And so eukaryotic cells are ones that have a true membrane-bound nucleus. Prokaryotic cells are those that evolved before there was a membrane surrounding the nucleus. Now, do both kinds of cells have DNA? Yeah, absolutely. I mean, think about the basic requirements that you need to be a cell. You've got to have cytoplasm or protoplasm. You've got to have a membrane. You've got to have ribosomes. And you've got to have genetic information in the form of both DNA and RNA in prokaryotic cells. What are some other differences in prokaryotes and eukaryotes? Got 2 fundamental ones so far. Size. Okay. Prokaryotic cells are much smaller. We'll take a look again at some images that give you relative sizes of various cells you're familiar with. But prokaryotes are in that micrometer world of measurement. What else?
Well, on a genetic level, how they transcribe and translate their DNA is a little different.
Yeah, there are some small but meaningful differences in how some of the processes of transcription and translation take place. When we refer to cells and their genomes, we use the terms oftentimes haploid and diploid. Which of those does not refer to a prokaryotic genome? Can a prokaryotic genome be diploid? No, they have typically a single chromosome, usually circular, and so each copy of a gene is only represented in one copy, so it's like they just have one allele. And so there's not that counterbalancing issue of dominance and recessiveness when you talk about prokaryotic genetics. What else? Do prokaryotes have mitosis and meiosis?
They don't have meiosis.
They don't have meiosis, definitely, because they don't form gametes. And technically speaking, they don't have mitosis. They don't form a spindle apparatus. Instead, when they divide by that binary fission, the cell membrane actually plays a role in the segregation or separation of that chromosome so that each cell gets a duplicated chromosome. And there's another reason why you really can't have mitosis in prokaryotes. What does mitosis refer to? What does it mean? Is mitosis equal to cell division? Are those two terms synonymous? No, mitosis is nuclear division. Cell division in eukaryotes consists of both nuclear division and cytoplasmic division, cytokinesis. Why can't prokaryotes have nuclear division? They don't have a nucleus. Okay. Which tend to have faster growth rates? Prokaryotes. We talked about the concept of generation time. Why is that the case? Why, in general, do prokaryotes have a faster growth rate, a shorter generation time than eukaryotes? They are simple. They don't have organelles that have to divide. That's true. But there's another more kind of fundamental reason. Gets back to one of the characteristics we just talked about.
Their high metabolic rate.
Okay. They have a high metabolic rate. That's one of the reasons why they can divide rapidly. Why do they have a high metabolic rate relative to eukaryotes?
I was going to say something.
Pardon me?
I was going to say something different. I don't know why they have a high metabolic rate.
Has to do with their size and their small size. What is the issue here that allows prokaryotic cells to support a high metabolic rate?
Surface area to volume ratio.
Good. Surface area to volume ratio, okay? Remember, as you get larger and larger and larger, your surface area to volume ratio becomes more out of whack because your surface area is increasing as the size of your square of the size increase, and volume is increasing as the cube of the size increase. So at some point in time, if a cell gets too big, it doesn't have sufficient surface area to sustain the activities of the volume. The surface area to volume ratio gets very unfavorable. What's surface area? Well, basically it's membrane. Why do you need a lot of membrane surface? To bring nutrients in, to get rid of wastes. And so if you have a higher metabolic rate, having a good surface area to volume ratio is really advantageous and really important. So here's a slide that is actually a few slides into the PowerPoint that describes and summarizes some of the differences between prokaryotic and eukaryotic cells, and those are things that you need to be familiar with. What I want to do, whoops. So a couple of these slides just show the relative size of various organisms that you may or may not be familiar with. Going from a human hair to ragweed pollen. You may know what a lymphocyte is? It's one of your cells. It's a white blood cell. It's a white blood cell. It's involved in your immune system. Here is a eukaryotic yeast. Here are some red blood cells. And then E. coli, Staphylococcus bacteria, Ebola virus. So this gives you kind of an idea of the size, magnitude differences we will encounter as we look at various cell types, both prokaryotic and eukaryotic. And that's one of the things that we're going to be doing in lab next week is we're going to be looking at different types of cells. We're going to be looking at a paramecium, for example, as an example of a single-celled eukaryotic cell. We're going to look at some red blood cells. And then we're going to look at several types of bacterial cells. So you can not only see relative size comparisons of them, but you can also get an idea of what some of the basic morphological forms the bacterial cells take. So we'll talk about concepts like bacillus and cocci and spiroem. Bacterial cells are fairly diverse in terms of their basic cell shapes or morphology, but if you break it down, there are basically three general patterns of cell type structure in the bacterial world. There are some variations of those, and there are some subclasses and things like that, but the three basic patterns are cocci, bacillus, and spiroem. Cocci are the circular, more or less circular bacteria. Bacillus are the rod-shaped bacteria. and spirillum have some type of curvature to them. And here you can see that there are several different variations of the kind of spirillum form here. But when we look in lab next week, we're going to be looking primarily at these three morphological types: cocci, bacillus, spirillum. Bacterial cells tend to oftentimes stick to one another following the process of cell division. And that will generate additional morphological patterns. So for example, here we've got a simple cocci, and if this divides in one plane to form two cells, and those two cells adhere together, then this particular arrangement is called a diplococcus. One of the things that you will see is that as you start going through Berge's manual and trying to identify your unknown, and you start becoming familiar with genus names of groups of bacteria, that many times a genus name is derived from the morphological arrangement of the cells that make up the species within that particular genus. That's one of the first things that early microbiologists first started to notice about different species of bacteria, is that some of them had different shape. So for example, there's a huge big genus of bacteria, many of which your unknowns will fall into, which is the genus Bacillus. And so these are going to be these rod-shaped, kind of sausage-looking like microorganisms. There is a genus called Diplococci. If the cells continue to divide and form long chains where the cells are joined together, then that particular arrangement is referred to as a strepto. In this case, since they're cocci, this would be Streptococcus. Okay, and that is a genus name as well. Sometimes cell division doesn't occur in just one plane, and instead of just forming chains of cocci, we will form various packets or clusters of cells. One common arrangement is a group of four, or a tetrad. So four cocci joined together. Here is another grouping of somewhere between 8 and 64 cells called a Sarcini. And that is actually a genus name too. There is a genus Sarcini. If the cells divide irregularly in multiple planes, they can form irregular clusters, and those irregular clusters are typically referred to with the prefix staphylo. And so staphylococci are these grape-like clusters of bacterial cells. And the number is fairly indeterminate. They can be small clusters, they can be pretty large clusters. I'm going to skip that one. The spirillum one we will look at in lab more, but some bacteria have a wavy shape, kind of a helical shape. Sometimes it's very abbreviated so that those bacteria look just like kind of a little comma, for example, just kind of one twist in their cell morphology structure. Those are called Vibrio. Vibrio is the genus that cholera is found in. I've made some reference to cholera a couple of times when we talked about the history of microbiology. So it's pretty easy with a standard light microscope and some very simple stains that allow the cell to pick up the stain and therefore become kind of countered to its environment to be able to discern the basic morphological form of your bacteria. And that's one of the first pieces of information you're going to get about your unknown is what's its basic morphology. Because when we start using keys to identify these things, oftentimes one of the first dichotomies is based upon morphology. Is it a cocci, or is it a bacillus, or is it spirillum? And that will then take you to different parts of the key depending upon what your unknown happens to be. Word of caution. There are some bacteria that are little short rods that when you look at them, they really look more like cocci. And they're in this kind of netherworld between cocci and bacillus, and they're called a coccobacillus. So when you're identifying your unknown, you have to really make sure that you properly identify that morphological shape, because in keying, a coccobacillus keys as a bacillus. So if you see something that looks kind of like this and you go, my gosh, is that a cocci or a bacillus? If you get that wrong, that can take you into the wrong part of the key and really mess you up. I'll come back to that in a second. Another type of basic information that you will get about your unknown early on is something called a gram stain. And a gram stain has to do with cell wall structure. And there are two basic categories of bacteria. There are gram-positive bacteria and gram-negative bacteria. And we'll talk later on when we talk about cell walls about why some cells turn gram-positive during the staining process. Gram-positive cells are purple following the gram stain, and why some cells are gram-negative, they stain pink during the gram stain. But this is another really important aspect of your identification, because oftentimes this is the other very early determiner of where you go in a key. Is your unknown a cocci or a bacillus? Is it gram-positive or gram-negative? Because these two are so important and so early on in the key, and sometimes can be a little bit tricky to identify, I will allow you to check with me when you start working with your unknown and verify the morphology of your unknown and the gram reaction of your unknown. Because I don't want you to get off on just a totally wrong tangent early on. There are some bacteria, for example, that look like they're kind of, some of the cells look gram-positive and some look more gram-negative. It's called gram-variable. So if you've got one of those, what do you do? So I would encourage you to take advantage of that. Surprisingly, I've had some students in the past who have never asked me to verify their unknown. And some have gotten it wrong and some haven't. But that's basically free information. And you don't get much free information from me about your unknown. So I just wanted to give you a heads up about that. Okay. So, prokaryotic cell structure. What kind of things do prokaryotes have? You're going to be really underwhelmed next week when you start looking at the bacteria and how unimpressive they are and how little detail you can see about them. I mean, the cocci are just little circles. The species of cocci we look at will be Staphylococcus, and so typically they'll be jumbled together into these grape-like clusters. The bacillus tend to form more individual rods, but sometimes they are found in chains, so we would call that a streptobacillus if they were in a chain. But you can't see much detail. I mean, you really need an electron microscope to really start getting into some of the sophisticated detail of prokaryotic cells. There is one other thing you can do, however, to facilitate gaining information about the structural aspects of your organisms you're looking at, particularly unknown. And that is, there are some stains that you can do that will allow you to differentiate between, does the cell have this or does the cell have this? The gram stain being a good example. Okay? But there are stains that allow you to determine, do your cells have endospores, for example, or do they not? Do they have a structure called a capsule? Do they have structures called granules, including one called a PHP granule, or something called a glycogen granule? So there are some things we can do with your unknowns, even with just the light microscope, by judiciously using stains to help us visualize the cells better and start to see some of this basic structural information. And that particular information will be important in identification of your unknown. That's what we're really going to spend a big part of the first half of lab doing. Is first thing you'll do is you'll start learning some basic sterile technique. Because once you get your unknown, you have to be able to transfer your unknown from the media that I gave it to you on and some fresh growth media or some kind of test media or a slide so you can do a stain on it. So we're going to do practice the first couple of weeks some of those sterile techniques. But then we're going to get into some of these staining tests, and we'll do the endospore test, we'll do the gram stain, we'll do a PHP stain, and an acid-fast stain. And you're going to start racking up some information now about your unknown, which will be useful in terms of its identification. After the first section of labs, after our first lab exam, we're going to shift over now and start gaining some information about your unknown from a metabolic standpoint. And that'll involve putting your unknown into various growth medias, like fermentation broths, and see, can they ferment this sugar? And if so, what kind of byproducts do they produce? Do they produce an acidic byproduct? Do they produce a neutral byproduct? And that's going to give you a whole new set of information. Some of you are going to be kind of frustrated when you do the staining reactions because your organism is always going to go negative. It's not going to have a positive result because it doesn't have this structure, it doesn't have that structure. And you're going to be going, man, I'm not learning anything about my unknown because it's giving me all these negative results. Well, in a dichotomous key, a negative result means just as much as a positive result. Because if it's got the structure, you go this way. If it doesn't have the structure, you go this way in the key. So don't get frustrated when you start getting a lot of negative results. Many of you will have unknowns that will fall into this group of bacteria called enterics. And these are gut bacteria. And these guys typically are gram-negative short rods. And when you do the staining reactions on a lot of these enterics, they're going to be negative for endospores. They're negative for this and negative for that. But that is meaningful information to know that it doesn't have some structure, just as it's meaningful information to know that it does have some structures. Okay, so here is kind of a composite of a prokaryotic cell showing some of the types of cell structures that might be present given a particular species of bacteria. Now, this is kind of a composite. It kind of gives you all of the different possibilities. Typically, most bacterial cells do not have all of these cell structures. So, for example, a lot of bacteria are not flagellated. They don't have these structures of motility, the flagella. Many bacteria lack this structure called the capsule. Many bacteria have inclusion bodies, but the different types of inclusion bodies can differ from one species of bacteria to another. So what we're going to do first is we're going to kind of just go through the laundry list of all the possible things that a bacterial cell might have. And one of the things we're going to concentrate on is how can you determine whether a bacterium has that particular structure or not? So we'll fold in, as we talk about bacterial structure, things like an understanding of how the gram stain works, or what's the basis for something being acid-fast versus non-acid-fast. Here's a nice little organizing slide that you can use to kind of start breaking down prokaryotic cell structure. So typically speaking, when we look at prokaryotic cells, we look at structures in terms of kind of three general categories. External structures, those would be ones that would be found external to the cell membrane and the cell wall. And they would include things like appendages, the glycocalyx. Those words probably aren't that familiar to you. Some of these things you might have heard of before. Flagella, obviously, but hyli and fimbriae you might not have heard of before. Capsule, slime layers. As we move inward toward the cytoplasm of the cell, the next set of structures typically constitute what we call the cell envelope. And that includes the cell wall and the cell membrane. Bacterial cells have to have a membrane. Can't be a cell without a membrane. Virtually all bacterial cells have a cell wall. There are a couple of examples of bacteria without cell wall, and we'll talk about those. We'll spend a fair amount of time digging into the cell wall structure, particularly in regard to this concept of what makes a cell gram-negative versus gram-positive. And then once we get into the interior of the cell, the cytoplasm, then we start talking about structures like ribosomes, inclusions, the nucleoid, the actin cytoskeleton, and the endospore. So this is a slide that I think is worth maybe even printing out and kind of keeping for reference if you don't have PowerPoint slides or printouts with you. Because as we're talking about something, I think that it's good to put it in kind of the context of this organization, so you kind of know where in the cell that you're talking about things. So basically what we're going to do as we go through our tour of the prokaryotic cell is kind of start here and just work our way down toward the interior of the cell. So we'll start with appendages first. The first appendage that we're going to look at is the flagella. You've all heard of flagella before. What's the function of a flagella? Motility. Okay. What's a good example of a human flagellated cell? Sperm in males. Okay. Now, in eukaryotic cells, there is another type of structure of motility besides the flagella. What's that? Cilia. And we have ciliated cells in our body. Where's the place we have a lot of ciliated cells? Got trachea particularly. Okay. Flagella and cilia, if you remember back to cell biology, are very similar structurally. And they have kind of a common organizational motif that we refer to as a 9+2 arrangement. Remember that term? Remember what the 9 and the 2 refer to?
The 2, I think, is like in the middle of the centrifugation.
It's in the middle of the flagella or the structure, and then there are 9 of these circular things. What are these circular things? Those are microtubules, remember? Okay, so we have a 9+2 arrangement of microtubules. And that there's all that interesting interaction between the microtubules and some other proteins called dynein, where ATP phosphorylation of the dynein protein causes those proteins to grab onto the microtubules and bend them, so that movement occurs in the flagella of eukaryotes by these flagella moving back and forth in kind of a whip-like fashion. Does that sound familiar at all? Okay. Well, prokaryotes have flagella too, at least some do. They do not have cilia. Okay, just flagella. But the organization of a prokaryotic flagella is fundamentally different from that of a eukaryotic flagella. In the prokaryotic flagella, the flagella consists of a single hollow tube of protein. Not 9+2 of those, but a single hollow tube. And that particular protein is called flagellin. We'll come back and look at the organization of the prokaryotic flagella in just a second. There's a couple of other things that I need to tell you first before we get there. If cells are flagellated, then there are different arrangements of flagella depending upon the species of bacteria that you'll find. The 2 basic categories of flagellar arrangement are polar and paratrichous. Which one of those arrangements, of those 4 images up there, do you think would be paratrichous? A, B, C, or D? D is paratrichous. Why do we know that? Para means around, like perimeter, okay, distance around. And so these particular flagella are found around the perimeter of the cell. They're kind of sticking out like hairs all over the surface of the cell. In polar arrangements, what do we find? We find flagella at either one or both ends, but not completely surrounding the cells. So in polar arrangement, it's at the ends of the cell. Now here they have three different types of polar arrangement that are illustrated. If you have a single flagella coming out of just one end, that is referred to as monotricious. That's easy to remember. Mono means one. Some bacteria will have a tuft or a bundle of flagella all protruding from a single end of the cell. That one's not so intuitive. That one is called lophotricious. Here we've got polar flagella where they're found at both ends of the cell. And that particular arrangement is called amphitricious. Well, could this be important diagnostic information about a cell? And is this flagellar arrangement species specific? And the answer is yes in both cases. So if you could get nice pictures like this of your unknown, you'd have some meaningful information that would allow you to help identify what your unknown was. But there's a problem. And that is flagella are really, really tiny. They're very, very slender. And the only way that you can actually see flagella using a standard light microscope is to do a special staining procedure called the flagella stain. And the flagella stain is really tricky. Because what you're doing actually is you're doing a staining procedure that involves selectively precipitating chemicals so they stick to the flagella, but nothing else. They basically coat the flagella and make it bigger so you can visualize it. So this is an example of a flagellar stain, and this particular bacterium right here has been properly stained, so all of those paratricious flagella have had chemicals deposited on their surface, and they're big enough now so you can see them. They've artificially been made bigger. You think this is an easy stain to do? No, it's horrible. We used to do it in lab, and it takes a whole lab period, and of a lab section this size, probably one person out of 20 would get a meaningful result. Your slides have got to just be absolutely clean. The chemicals have got to be, really pure. And it just wasn't worth it. It wasn't worth the time or effort. However, there will be instances where it is important to know, as far as keying, at least whether your flagellar arrangement is polar. It's not particularly important to know whether it's monotricious or amphitricious, but that it's polar, or whether it is paratricious. So this is instance 2 where I will be willing to provide you with information should you need it. Now, there is a test we can do to determine motility of an unknown. And if your organism's not motile, it doesn't have flagella, so you don't need to know arrangement, obviously. But if you are motile, then I will provide this information to you in terms of this idea of polar versus paratricious. Okay. So here's a flagella. Flagella is basically this bent protein tubule that has a series of other proteins associated with it in a ring-like structure. The three basic parts of a flagella are typically considered to be this ring-like group of proteins right here, called the basal body. And then the flagella extends into this hook region where it bends, and then the long, elongated part of the flagella is called the filament. The filament makes up the vast majority of the length of the flagella, and basically it's a little hollow tube of protein. So it's kind of like that cardboard paper towel insert in a roll of paper towels. The flagella actually grows by transporting flagellin subunits from the cytoplasm of the cell. So the cell's got to have the genes to make flagellin, and then it makes the proteins on ribosomes. And then it transports those flagellin proteins through the hollow tube of the flagella, and it assembles and grows at the end of that particular filament. So if part of it gets knocked off, they can kind of regrow again. The basal body is basically the motor of the flagella. And one of the things that you'll see that's fundamentally different about the flagella in prokaryotes and eukaryotes is, remember, eukaryotic flagella do this. As those microtubules and dynein proteins change relationship to one another as they're phosphorylated and dephosphorylated, and the microtubules bend, and they bend, and they bend. Okay? The flagella in prokaryotic cells works kind of like the prop on a motorboat engine. It rotates this way. It doesn't do this, doesn't go back and forth. It rotates round and round and round. And so this basal body right here kind of represents the motor, and then the hook and filament represent the prop that is spinning around. What powers this? This is an ATP-dependent process, and ATP is used to establish hydrogen ion gradients. And a hydrogen ion gradient in the vicinity of the basal body is basically responsible for the motor of the flagella working. That's about all I'm going to say about it. It's fairly complex biochemically what's happening. One other thing that you can see is that we've got two different representations of flagella. They both look kind of the same, but down in this basal body region down here, you see that there are kind of rings of proteins here that look different from the rings of proteins here. There are two basic rings of proteins that make up the basal body in this cell type, and there are three basic rings of protein that make up the basal body in this cell type. This cell type we'll see later on are gram-positive bacteria. This cell type right here is gram-negative bacteria. So there is a little bit of difference structurally in the flagella of gram-positive and gram-negative, but they work pretty much the same.
Did you say the two proteins was the positive?
Yeah, two for the yeah. This is gram-positive on the right over here. Okay. And once we go through, Kenzie, once we go through cell wall structure, you're going to immediately be able to recognize this outer second membrane right here called the LPS layer. That's associated with gram-negative bacteria. The gram-positive bacteria don't have that outer membrane, that outer LPS layer. So as you review for your first exam, that'll become pretty obvious to you. Okay. Flagella can rotate in two directions, counterclockwise or clockwise. If they rotate in a counterclockwise direction, that particular rotation is called synchronous rotation. Everything's in sync. Everything's working right as it's intended. And this generates a type of movement that we call a run. And a run tends to be directional. However, if the flagella rotates in a clockwise direction, then that's considered to be asynchronous. And basically, that's kind of an uncoordinated flagella. That gives you a type of motion called a tumble. And tumbling motion is random. It's kind of erratic. drunk stumbling down the street. Okay. What determines whether the flagella rotates in a synchronous fashion, giving you a run, or an asynchronous fashion, giving you a tumble? Directional movement versus randomly wandering around. Well, associated with the flagella in the membrane of the cell are some receptors. And these receptors respond to a variety of things, including chemical stimuli. This run or directional movement is oftentimes a positive chemotaxis. A substance is attracting the cell and stimulating it to move toward that substance. What might be positively chemotactic? Maybe a food source would be. Okay. Tumbles or random motions tend to be associated with a negative chemotaxis. Things randomly moving away from the stimulus, not toward them. So again, a taxis is a response. In this case, a movement response. Chemotaxis is response to chemical stimuli. Positive chemotaxis is attraction. Negative chemotaxis is repulsion. Now, there are other stimuli besides chemical stimuli that can influence the rotation of the flagella. Some bacteria are also phototactic. They respond to light. And I think you can see the obvious advantage of being attracted to a light source if you were a photosynthetic bacteria. So substances that stimulate counterclockwise movement of the flagella tend to result in runs, which tend to be more directional movements toward the stimuli. Things that are repulsants, if you like, negative chemotaxis, tend to cause the flagella to just not work properly, and the bacteria is kind of stumbling around. And so we get this kind of random motion right here, the tumble, and that oftentimes results in the bacteria moving away from the stimulus. So bacteria are able to chemically respond or phototactically respond to environmental factors as a result of the interaction of that stimulus with some type of a receptor associated with membrane, which then influences rotation of the flagella. Alexandra?
I guess, is it like a chemical that's being sent to the cell that's telling it?
This is something probably in the environment, or maybe something even that the cell is producing. So if the cell is producing a toxin, for example, and that's diffusing out into the environment, then that toxin would tend to stimulate the cell to move away from that substance, which could be poisonous. So these typically are environmental substances that the cell is then responding to.
See, it's completely about negative chemotaxis and random motion.
Mm-hmm.
Would it be like beneficial for it to move directional when there's danger or when there's repulsion? Because if it's random, there are chances that it might.
Yeah, I see what you mean. I mean, there's really the way things have evolved. There's no real mechanism associated with being able to move directionally toward a stimulus and away from the stimulus. And so your choices are basically generally moving toward the stimulus. I mean, if you start to veer off a little bit, then your concentration of your attractant gets less. And then if you kind of end up veering back toward it, the concentration gets higher. So it's not just even a straight directional movement. It's kind of a wandering, but generally toward it. The other one is just kind of a blind stumble, and, yes, you might blindly stumble into the repulsant, but generally speaking, you're probably going to be less likely to move toward it if you're just kind of blindly moving around. So it's not a greatly efficient mechanism, but it's the way it evolved. It works reasonably well. Good question. There are some other types of flagellar arrangement. One that we're going to come back to and talk about a little bit, and I just want to introduce it right now, is a type of flagellar arrangement called a paraplasmic flagella. And these are flagella that are wound around the cell, almost kind of like a belt around your waist. And as those flagella expand and contract, they create a corkscrew type of movement. And one of the species or types of bacteria that have these paraplasmic flagella are a type of bacteria called spirochetes. And spirochetes can actually burrow into tissue because of the motion of these paraplasmic flagella. It's really almost like a corkscrew turning into a cork. Syphilis is a spirochete, and the bacteria that causes Lyme disease is a spirochete as well. And that's kind of part of their pathology, is their ability to burrow into tissues using these paraplasmic flagella. Okay, probably a good place to stop. We pretty much covered what we want to cover so far about flagella. So the next thing we do is we'll look at next Tuesday at some other appendages, hyli and pimples. So, have a good weekend. Come back refreshed. Okay. Chemotactis is actually the cellular response. It's a movement, tactic means movement. So it's a movement in response to a stimulus. So bacteria can show both positive chemotaxis and negative chemotaxis. They move toward stimulus or away from the repellent. So chemotaxis refers to how the bacteria behaves. No. Now when you break down, the word chemotaxis, the response in the case we're talking about now is a movement. Okay.
So it's the movement of the flagella.
Yeah, it's a movement of the flagella, and it's a response to a chemical stimulus. So if you have something that is positively chemotactic, those bacteria are going to be moving toward some kind of a chemical stimulus. If a bacteria is showing negative chemotactic behavior, it is moving away from some kind of stimulus, in this case a chemical stimulus.
So the stimulus is picked up by these receptors,
Yep.
and then that depends on if it's positive or negative.
Mm-hmm. Mm-hmm.
And then you were talking about the 9+2, and then you said the single hollow tube. So this is the, were you saying this is like the basal body part, or like?
Well, I mean, in the eukaryotic flagella, the basal body has actually, it's a little slightly different in this structure. I think there are 3 bundles in here, but then the bulk of the flagella consists of these 9+2 filaments. It's actually on a eukaryotic cell. If I've got a eukaryotic cell, and then here's the flagella sticking out here. Okay, if you do a cross-section of this flagella, this is just an extension of the membrane. Okay, this is just an extension of the cell membrane, and then there are microtubules packed into here. So if you did a cross-section through here, this is what the flagella would look like. There would be these 9 bundles on the outside, and then these 2 in the middle. Prokaryotic cells, not nearly that complicated. They just got basically one little microtubule sticking out.
So this is all.
That's all eukaryotic. Yep. And it's kind of unfortunate in some respects that they use the same word for both a prokaryotic and eukaryotic flagella, because they're so different structurally.
That
Okay.
makes sense. Thank you.
Good.
And then I have the form. I just forgot it in my room this morning. So
Okay.
can I just get it to you bring it to class on Monday? Is that fine?
On Tuesday. You know, that's
Tuesday.
fine.
I'm sorry.
Yeah, well, we got to turn it in Tuesday because
Yeah.
that's last day to have.
Okay.
Okay?